I'm Ethan Stern. I'm a cell biology neuroscience major.
I'm Joe Ibrahim, and I'm a cell biology and neuroscience major.
I'm Anish Vagela, and I'm a biomedical engineering, and we are the genetic engineers at Rutgers Society. So the genetic engineers at Rutgers Society is a group of students that has a passion for genetic engineering. We started 2 years ago, basically in a room of about 20 students that wanted to do iGEM, wanted to compete in the premier synthetic biology competition hosted by MIT.
The iGEM teams really function on the concept of synthetic biology. It's a new approach of research work that combines science and engineering at the same time. Synthetic biology concentrates on not making the product, but how to make the product. We alter pathways that already have been done before, pathways to make biofuels in bacteria.
One of the really unique things about our organization here at Rutgers is that we're completely independent of any researcher who is in charge of us directly. So we design all of our own experiments, so that's really huge. You don't get that opportunity really ever as an undergraduate to really design your own experiment and follow through with it.
I was taking a class called Introduction to Molecular Biology and Biochemistry for Research. It's a class taught by Dr. Andrew Vershawn at the Waxman Institute of Microbiology. He really taught us everything that we need to know about synthetic biology and genetic engineering, and got us as undergrads to be a little more experienced with the research.
He'll teach you anything from basic pipetting techniques to how to set up a PCR. And from this course, students that worked and did well in the course were able to approach him and request lab space and mentorship for this project. The professors that have done research oftentimes are very encouraging and shed light on our projects and try to give as much advice as they can for us.
Over the past 2 years that we've been in an organization, we've really come to understand, about more what genetic engineering and synthetic biology actually are. We're really going around, we're looking at more scientific papers, we're trying to find more creative influence to create better projects.
The name of my research project is 1-butanol production in E. coli. So this year, Rutgers iGEM decided to take an approach on renewable energy. By doing this, we decided to produce butanol, which is a biofuel, in E. coli. This bacteria can produce high amounts of this fuel and can potentially be used to fuel vehicles, lawnmowers, anything that's basically gasoline powered. So for example, if you were to have a vehicle that is capable of running on ethanol, butanol is highly similar to ethanol, and since that similarity exists, you would be able to run these vehicles on the fuel that we are producing in the bacteria. Biofuel production, namely butanol production, was a heavily researched area that we wanted to expand upon. The fuel industry is very oil dependent, and as we strive to find ways to reduce our dependency on oil, bacteria such as these that produce these fuels can potentially replace what we have today in the oil industry.
ETCH-A-SKETCH as a project itself is forming a biosensor within a bacteria, or taking bacteria and turning it into a biosensor so that it can detect anything that you want it to. What we're doing is we're laying bacteria on a board, and we're using a certain intensity light, and then we're shining that at the bacteria. And using the circuit, the bacteria is going to absorb the light and trigger the red fluorescence protein, and it's going to shine back as red. So essentially, as a way of expressing this project without having to use toxic materials, we're sketching out using bacteria. If a body of water is suspected to have toxic materials from a factory or from an incident that has happened before, just releasing this bacteria, this harmless bacteria, into the water can detect, using fluorescence, what is actually in this water and what level of toxicity is actually in there. So at the competition, what we're planning to do is to take this bacteria, lay it down on a board, and using the blue light pen to shine the word Rutgers on it. Considering the efficiency of our project, it should shine back with fluorescence, and the word Rutgers should shine back from the board. And that's how the name ETCH-A-SKETCH came about.
The bacterial cellulose synthase project is about taking a gene that makes cellulose from a bacteria and putting it into E. coli. However, this project has already been done, so what we're doing is we're taking 2 other genes found in E. coli and overexpressing them, which means that we're going to make a lot of copies of them. And we're theorizing that when we overproduce these 2 genes, we get a lot more cellulose. And when we have more cellulose, it's another cheap outlet for it. It's actually the main component of paper, cardboard, textiles. It's main component of plants. It's very strong fiber. It's partially digestible if you really need food. Bacterial cellulose is actually considered to be a better product than plant cellulose. It's considered to be more pure. It's considered to be, cheaper. The only problem is that we don't have an efficient method of producing it right now, and my project that I'm working on will hopefully fix that. One of the reasons I actually came to Rutgers is because I'm so interested in research, and Rutgers has a huge undergraduate research population, and a lot of different professors are willing to work with students. And this just seemed like a really great opportunity to really get into the down and dirty of how a lab works and really design my own experiment.
Undergraduate research is great in terms of networking, leadership, and just learning and creativity in general.
The biggest thing and biggest concept of research is really just having an open mind, being a little more creative, and really just looking out there for what's possible and what can be done.
Research for me, it just gives me this type of excitement. It gives me this joy of, going in the morning and getting my results and, seeing how things work. And it's really interesting to see how these bacteria respond to different things. And I really just enjoy working with the team that I'm working with right now, and it's just been a great experience overall.
We want people to be really driven. We want people to be creative. You don't really have to have too much lab experience. We can teach you everything you need to know. It's really just about, being motivated. If any student interested in joining GEARS or participating in iGEM, you can reach us at rutgersigem.com. We're always looking for new students to help us out. We're looking for different creative, driven individuals who really want to learn about how a lab is conducted and learn about synthetic biology and genetic engineering.